All right, so MycetOS meeting in the 26th of March, 2024. And the agenda is online. I guess, I mean, if you guys are all right, did you want to talk about any new biological data first? Because I know it crosses all series, but then at least we know what we're talking about. Any new in vitro evaluations, for example, that we need to think about?
Yeah, I made a slide.
Marijn made the slide. Where is the slide?
Yeah, in my.
Yeah, is it on the V drive? Then I can access it as well.
All right, fine, great. Yeah, it'd just be useful to know.
Yeah, because Marijn is currently doing the screenings.
All right.
So she has an update, and Ma also has an update on Series 5.
Oh, awesome. Okay.
And I think it's in results. And then, yeah, the results MIC50.
Okay. Then I will share the screen. It's opening the slide.
Great.
Hopefully.
Just say congratulations on the grant, Wendy.
Yeah, thank you.
That's
it helps
awesome.
helps us to build MycetOS further. So
no, for sure. Yeah, it's great.
Because we're going to use the open science not only for the drug discovery, the drug making, but also for the model making.
Yeah, absolutely. Yeah.
So let's see. This one big. This one small, and share. Okay.
So yes, I made a summary of my results from the MIC50 where I'm currently working on. You've seen the table, the MySCO, the other IDs, the series, and the MIC50 in micromolar. These are the 50 compounds I did the MIC50 on. Ma and Wendy, we selected and analyzed these compounds to see which compounds are good to test in the larvae, and they are highlighted in red. They are from series 3, and yeah, we think it's these compounds are the most interesting ones to test, I think.
Yeah, the problem is because Ma is in the end of her PhD, and Marij is also finishing before the summer. We don't have much time this larvae season, so we thought, well, we already tested series 1 and completed the manuscript, so those few molecules still left, maybe not worth to focus on too much. Series 6, all the compounds we tested so far were not active in vivo, although they were active in vitro. But we don't have data from Series 3 yet. So we thought, let's screen Series 3 to see if it does do something in vivo. Let's start with that and see how much time we have left in the season to do the other things too.
Right.
Yeah, that was a little bit the rationale behind it.
Okay. The oxyconazole nitrate,
yes,
which is the most potent compound, is that active in the in vivo model?
I don't think we tested oxyconazole yet.
You mean
this one?
Yes.
No, we didn't test that yet.
No.
Because that's a Series 6, and the other ones were not active. So
yeah. It's just funny that I mean obviously it's an antifungal, right? So you just wonder if you'd expect that to be active. Would you expect that to be active because it's a known compound?
Yeah, and it depends. Belongs it really to the azoles, or is it a different series? Because all the azoles we tested so far, they were very disappointing in the larvae model, as well as in the mouse model later. So if it's an azole, I don't expect too much of it, because even ravuconazole, although it prolonged larval survival, it was not that that promising. We've seen better compounds.
Right. I guess it's just, you know, I mean, it's a funny disconnect that we have this potency in vitro and not in vivo. And I guess if you were thinking about a completely new chemical structure and motif that we'd thought up for Series 6 that just didn't seem to be compatible with the in vivo model, then that would explain it. But if you're dealing with oxyconazole, which is an actual antifungal medication that's on the market, wouldn't you, which has the same motif in it, right? Would you expect it to be active in the model? I mean, should the model pick it up if it's
not necessarily, because itraconazole is, of course, the drug currently used to treat the mycetoma patients, but in our model, it doesn't do anything. In the mouse model, it doesn't do anything. And even in the patients, after 6 months of therapy, the grains are still viable and you can grow them out quite easily. The only thing itraconazole does is encapsulate the lesion very well, makes it easier to excise. And that's the difficulty here as well in the patients. They always combine medical treatment with surgery. So it's difficult to say which effect is really from the drug by itself. And none of the drugs currently in use are able to cure it without surgery.
Right.
So that means that none of the drugs currently in use are actually active enough to do this on their own, while the bacterial variant of mycetoma can be cured without surgery.
Right.
So that's why I'm leaning more towards finding novel targets, because Series 6, like Series 1, is targeting CYP51.
Yeah.
And the most potent azoles didn't do it. The compound we tested so far for Series 6 didn't do it.
Yeah.
And the azoles themselves didn't do it. So maybe CYP51 is not the most ideal target to look for when we want to penetrate the grain. The grain is a biofilm. And we also know that for other fungal infections, the azoles are not a good target for biofilms.
Yeah.
So they cannot either inhibit or either penetrate, or when they inhibit, the effect is not good enough to destroy the biofilm. And I hope that if we go to a complete new target, like for instance Series 3, that we might be able to do that.
Yeah, for sure. I mean, that's one of the reasons. I mean, that's the primary reason for changing the series and, you know, introducing new motifs so that we can get outside of what we're currently doing, for sure. For the what is that? The in the middle of the right hand table, you've got one of the Series 1 compounds, which is 0.125. I forget if that's about as potent as we saw, I think, for Series 1. Okay. And I don't know if that compound, I can't remember what that compound is, but I guess we are about, well, we're hopefully getting very close to submitting the Phenormal manuscript. These molecules should be added to that, and we can certainly deal with that. I guess you don't see any particular, again, for the same reason, you don't see any particular reason to include that 0.125 compound in the in vivo.
We could include that particular one, but it would mean that we have to postpone again the manuscript, and maybe.
Sure. Yeah. Okay. Well,
that was a little bit the reason behind it, because we could try to include this one. That's not a problem. But it might not be that good to postpone the manuscript on it, because it has been, and it's needed for, I think, Dimitri's and Hang's thesis.
Well, so I mean, the point you're making though is that, I mean, the in vitro data are needed for the SAR, right, to understand, so sort of confirm the target that we're talking about, and hopefully it makes sense based on what we know. But the message from the in vivo is that despite the potency, we're still not impacting the disease in an in vivo model. And but given that all the molecules are actually quite similar, it would be unexpected if one of them suddenly did well in the in vivo model. So all of this is contributing evidence to the idea that we need to be looking at different targets.
Yeah, and we did learn that the log D was important to penetrate apparently the grain. Doesn't necessarily mean that that's the same for all the other series.
Sure.
But if we now have a series where we have more than a few compounds, it would be good to test if those which did prolong larvae survival also have a low log D or something which sticks them out. So it would be great to see if all the types of molecules which penetrate at least have a similar feature, so that we can use that in the design.
Yeah. Yeah. Okay. Yeah, no, it makes sense. I guess, yeah, series 3 is a new thing, so.
Yeah.
And have we, again, apologies for my memory, have we tested any of the series 5, series 2, the stuff that we've been receiving from other places, the aminothiazoles? Have we tested those in the in vivo model?
Well, the first molecule was, of course, tested in vivo. That was our hit. I think we did test a few, but none of them were active.
Three compounds from series 2 were tested in vitro larvae, but both against the Moderula mycetomatis and Phosphormispora snigilensis, but no one has had active action.
No, there was no. Although the original molecule was very active in the larvae, it was one of the most potent. But the derivatives we made so far, they were not active.
Okay. There's one there with a MIC of 50 of 2 down the bottom of the left-hand side. But again, if that's not particularly different from the potency that we saw for the previous compound, then there's not a lot of point in including that.
Yeah, and the same for the kinase inhibitors. That might be very interesting too. But yeah, at some point we needed to prioritize.
Yep. Yeah.
And we thought, well, let's go for series 3 first, and then see if we can, with the time allowed, add a few others too.
Yep. All right. Great.
And of course, it will be easier next year when the new PhD student has started. Then we have more hands to screen again. But yeah, for this year, it might be a little bit more difficult. And this one we selected, but we also screened the 6,000 compound library where new hits came from. And we thought maybe it's good to first test those too, so that we have new scaffolds to think of.
Sure.
And when you get new PhD student, or maybe in Sydney, then it's good that they can start with the new series.
Yes. Yeah, absolutely. A new target would be great. Yeah.
Yeah.
All right. So your timeline for the in vivo testing is, I guess, the summer.
Well, we tried the first larvae last week, and it seems, because the weather was fine, that they already were able to use the larvae so far. So we're going to plan to scale it up and start somewhere in April.
Okay, great.
Because that also gives Marij enough time to finish at least these things for her thesis, her bachelor's thesis.
Okay. All right, wonderful. So these results that you're showing on the screen, if they can be added to the master list and then color coded as new, then we can.
It will be added, and of course this is the MIC 50, so that means that it's the middle value of all the MICs for different strains, but we will add the individual results too.
Right. Wonderful. Okay.
Okay.
Great stuff.
Shall we continue with Ma? But then we jump immediately to series 5, or do you want to go first to the other?
No. Go for it. If you're sharing, go for it.
Okay, then.
This presentation aims to introduce the introduced compounds from series 5 from MycetOS. It's a small summary from 2022. 17 compounds were selected, were delivered, and 16 compounds were delivered at 2023. Those compounds were screened by me, and the other 20 compounds from series 5 were screened by Maria. I want to note the difference of Moderula mycetomatis and Phosphorus porous nigrolensis first, because they have different morphologies from this picture, and they're from they are from different orders. So we selected these two strains to screen series 5 compounds.
They are also the most common causative agents of mycetomas, fungal mycetoma. So
the two most common causative agents of mycetoma. So this is the MIC data and MIC50 data from series 5 compounds in 2022. Finally, 3 compounds of 17 compounds were active against Moderula mycetomatis with MIC50 value below 8 micromolar. From series 2023, 6 compounds were selected by MIC50 value below 8 micromolar. Then we tested all compounds against Phosphorus porous nigrolensis. We can see just 3 compounds were active at 25 micromolar. Then we test the MIC data. The MIC test was performed. Then these 3 compounds were active against Phosphorus porous nigrolensis with MIC data at 8 micromolar. So these 3 compounds were selected to do further in vivo larvae test. Then this is a small summary. In total, 33 compounds were selected. Then 9 compounds were selected from Moderula mycetomatis with MIC50 value below 8 micromolar, and 3 compounds active against Phosphorus porous nigrolensis with the MIC value. From these 2 branches, 3 compounds were active against both 2 strains. So these 3 compounds were selected to do in vivo test. Then at first, the toxicity test was performed. Both 3 compounds were toxic at 20 micromolar. We usually use this dosage. Then we use the lower dosage to do the toxicity test. Then both of them were not toxic at 10 micromolar. So we decided to do the in vivo test at 10 micromolar. Then we performed the larvae model against Phosphorus Moderula mycetomatis in the left and Phosphorus porous nigrolensis in the right. We can the data show the MYOS 333 and MYOS 334 prolonged Moderula mycetomatis infected larvae survival with a significant difference.
Yeah, but of course it's not that promising, but it did prolong larval survival.
Yeah, because 15 larvae were from one time tester. So I repeated 4 and 5 times, and they did prolong larvae survival. So yeah, but no compounds prolonged phosphomysporous neglections infected the larvae. Then we made the slides for observing the grain formation of this treatment. The left one is from Motorella mycetomatis, and the right one from the phosphomysporous neglections. The HE standing shown the inflammatory reaction, and the crocota standing was for the hyphae, the fungal hyphae, in black in the crocota standing. Yeah, and we can see there are no any difference between the PBS treatment and the Myers treatment, I think. Yeah. Then is a last summary. We selected three compounds to do in vivo test. Then three compounds were toxicity at 20 micromolar, but not 10 micromolar. Then two compounds prolonged larvae survival, but just against Motorella mycetomatis. Then this compound is from Maria site. Yeah, I didn't upload your new data. And as for just the one compound, Myers 527, I thought the MIC50 is at. I noticed just now. Yeah, at. So I think this one will be selected in this season to see whether it's active. It was active. It is active. Then yeah, we left a message to ask where to find the chemical information, and Andrea told us we can find the data in the OSNP database. So I go to the database, then search all these four compounds, and I found the log P. The log P data. So I updated them here. It looks like those four log P were below 4, below 5.
Yeah, but above 2.5.
Okay, then I also get their chemical structure. Then the first one, Myers 326, was not active in vivo, but Myers 33 and Myers 334 were active against Motorella mycetomatis in vivo. And we can see it's clear. Just this site has a different molecular.
No, it's more like this one. So if this was not active, this one had a chlor, and here you have brome.
Yeah. So yeah, even though we don't know too much of a chemical structure, we think it has interesting things, I think.
Yeah. So then, if that's the case, then if that really is a defining factor, this one should probably not be active in vivo. But I'm not sure if I can say it like that, because this one also has a very different side chain, of course.
Yeah, from you. Yeah, and there is a question. There is questions here. I plan to prepare the chapter for my thesis. So we need help for the compounds chemical information. So we know that all compounds from series 5, they were maybe from two groups, benzamidazole or benzox.
Yeah, original series 5 was supposed to be benzimidazoles, but the benzoxazoles were added, and these all look the same. So we just want to confirm these are benzoxazoles instead of benzimidazoles, correct?
Yeah, that's right.
Yeah.
Okay. So maybe we need more help for introduce what difference or what's or where is will be has this interesting
chemical features.
Chemical features, yeah.
Because yeah, the chlor we could see ourselves, but probably the rest is not that easy to see.
Yeah. Then yeah, that's all. Thank you for your listening.
Yeah, great. All right. So yeah, so there is this confusion, isn't there, about this series that we have two sub things in there.
Yeah, I was wondering, should we call it Series 5A and Series 5B?
Yeah, possibly. I mean, it's grown, right, because Andrea's contributed more molecules, and yeah, very possibly. And I think so Clementine has done a nice job of keeping the structure activity relationship diagram for the benzimidazoles quite up to date on the wiki. And that does sort of show things that if we were going to make more molecules, then what we might want to think about. But I don't think anyone has yet done the side diagram for the benzoxazoles and what we've learned from
Yes.
those and whether there's any overlap. So we're definitely going to need that. And that might tell us how similar things are and whether there are similar trends, and therefore whether we might want to consider them in the same paper or not. We just need the diagram, unfortunately. So that does mean that we've got to make sure we've got all of the activity data for the benzoxazoles together and have considered it all together. So I don't know who's going to do that, whether Clementine's able to do that or whether Andrea wants to do that. To sort of just ensure that in one diagram or something we have everything in one place, and then you can start moving things around and seeing what the pattern is. Yeah. It's
I will make it in the note of the agenda that that part is needed, and to see if either one of them would like to volunteer.
Yeah, we need an SAR diagram for the benzoxazoles.
Yes.
Yep. Great. That's good.
So then I think we stop sharing.
Right. Okay, great. Thank you for all that. In terms of the yeah, I mean the remaining things here, there was one thing on me chasing that compound 559, which we think has come from GSK Trescantos. Sorry, I haven't done that yet, but I will. That action's on me, can stay on me. I just need to reach out to them to say, where did this compound come from? Do you have any more of it? Are there other examples? You know, is there someone we should contact? Just to see if we can investigate things more there. So that's still on me. The plant extracts that we think we got from Nigeria, but we don't have anymore. It was the idea that the original people would try and resend those to your lab, Wendy.
Yeah, apparently they had also some new extracts so that they could be sent in one go. That was Bernard was going to go after that.
Right.
And one of the marks also with toxicity data, I just uploaded a column with toxicity data.
Oh, right.
And we're going to display at which concentration we know that it's not toxic.
Mm-hmm.
So for most of the compounds, it will be 20 micromolar. That's the highest concentration we test. But as seen for this series, so the benzoxazoles, it will be 10 micromolar. Because then we give a little bit more information than only saying toxic, yes, no.
Right.
Because at least the concentration where there's no toxicity will be shown.
Right.
For those compounds tested.
Is there a way of you know, imagine you're looking at the data in a year or two. Is there a way of linking the data somehow to a protocol so that we know what the value means, you know, how it was obtained?
Well, we do have the protocols, of course, how we do this. Probably we can put them somewhere, what it means, but yeah, it's a standard protocol, so we always do it the same way.
Right. I wonder. I mean, one possibility is to have a an issue or a thing on a wiki somewhere, you know, we have the sort of tech ops wiki where we talk about how things are done, or we have, or anything actually, where we could put a toxicity issue, and we could have protocols there and then just link to that from the master list so that people can see how the numbers are obtained. Just because I think that we're going to have toxicity data from a number of different places potentially, and we just got to make sure that the number is connected to a way in which the number was derived. And I know it gets fiddly.
Yeah, because that's also why I said toxicity in Galeria in the database, right. Because of course that's not the same as toxicity in a cell line.
Right. Yeah. Exactly. Yeah. Anyway, think about how you might want to do that. You know, I'm just thinking in a while, you know, we just need to make, so, you know, we've had toxicity data on in an antibiotics project from 2 different places, and you know, one at AstraZeneca and one at UCL internally. We just have to be really sure that we are, you know, connecting the number to the protocol. That's all.
Yeah.
Schools meeting was, sorry, sorry, did you want to add anything else to the things you were talking about? Because I was just going to summarize a few other things that we can summarize here.
No, because I think most of the other things are quite clear in the agenda, what needs to be done. So
yeah, I think so. And I think the people who need to deal with those things aren't here. So everything is progressing, but we do need other people to come and talk about updates, I think.
Yeah.
Yeah, and the DMPK measurements that Dundee did, I'm pretty sure that almost all those compounds are stuff that we ship from phenoromol series and from the ketoxime series. So the data are posted now. I had to edit the spreadsheet because there are other data in there from other projects. And it would just help if Dimitri and Hung could check that the compounds that were shipped were evaluated, because it looks like there are fewer compounds in that sheet than the number we shipped. So I just want to try and tie that all together. But I think that the data are relevant to those 2 series. So if there's anything from the phenoromol series in there, we could slip that into the paper if that's relevant. But I just want to check. And I think Hung has done a bunch of edits to the paper, which I will go and try and check in on. Now, I was asking for checking the experimentals done from his perspective, and also the stuff that he'd done on whether or not our results are, you know, aligned with the idea that we're hitting CYP51. There were some references put in, and apparently he's done that. So it's my turn to go in and check the paper.
Yeah.
All right. Other than that, yeah, I think that's everything, because everything else is assigned to other people.
Yeah, I'm still going to make. I still have to make the plate for Melissa.
Oh yeah, right.
Because I do have to check if we can share all the molecules. The molecules which were made for MycetOS probably are not a problem, but it's more the molecules from DNDi.
Right. Yeah, sure. I mean, I bet it's the same thing, right? But yeah, it's important to be clear. Yeah.
I see that Anthony joined.
Oh yeah. So yeah, we dealt with series 3 earlier, and some series 3 molecules are going into the in vivo phase, which is exciting. All right. Okay. Well, there's nothing further. We're nearly at the hour anyway, actually. But thanks for all the data from your lab. Congratulations on all those measurements. It's really helpful. And well, I think all the actions are clear, as you said. So if you're able to summarize anything there, but I mean, a lot of them are just remaining as they are, right? But hopefully we can.
Yes, but I will add the things we discussed.
Okay. All right. All right. Good. All right. Great. Well, very nice to see everybody.
Yeah, it seems like Anthony wants to say something, because I see his thing, his name going up and down, but I can't hear it. So
no. Okay.
Sorry. What series 3 compounds? The ones Peter made, or the NEU Curaxin ones are going into larvae?
The NEU ones, I think. From the codes I saw, it's the NEU ones.
Yes.
Okay, that's all. Thank you.
Okay. If you look at the recording, you'll see the codes if you want to check it.
Okay, thanks.
All right, so next meeting is after Easter, April 23rd. Happy Easter, everybody. Hope you have a nice break.
Yeah.
Bye, everyone.
You too?
Yeah. All right. See you.
Okay.
Bye.
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 Bye.